I'm Dr. Malarkodi, Professor and Head Department of Pharmacognosy, School of Pharmaceutical Sciences, going to discuss about the phytochemical screening of crude drugs for proteins, amino acids, steroids, and triterpenoids. First, going into the depth, we know what are proteins. Proteins are the essential biomolecules consisting of one or more long chains of amino acid residues. They are large macromolecules which play a vital role in growth and development, and therefore it is called as the building blocks of the body. They are the class of biologically important compounds with high molecular weight. Proteins are perhaps the most complex organic materials produced in nature. Plants build up their proteins from carbon dioxide, water, and minerals in the presence of sunlight, whereas animals derive it from eating plants. We will know about the functions of proteins. They protect the immune system, maintain the oncotic pressure of blood plasma, prevents the fluid from leaking out of blood vessels, and it helps in the transportation of ions, lipids, and other hormones. It is also important to identify the proteins to understand their functions, which help in characterizing the binding of proteins to other proteins, nucleic acid, or two small molecules. It also plays a major role in drug discovery. Test for proteins. To the plant extract, about 100 milligram, dissolve it in 10 ml of distilled water and filter it through Wattman #1 filter paper. The remaining filtrate is subjected to test for proteins. First comes the Millen's test. To 2 ml of filtrate, few drops of Millen reagent are added. A red precipitate indicates the presence of proteins. Millen reagent is chemically mercuric nitrate and mercurous nitrate dissolved in nitric acid and distilled water or acidified mercuric sulfate. Millen's test is given by proteins containing phenolic amino acids, and they detect the presence of soluble proteins. Phenolic group of tyrosine of proteins react with the mercuric sulfate in presence of sodium nitrate and sulfuric acid to give a red color. Here it is the diagrammatic representation of the Millen test. Millen's negative test, absence of tyrosine or phenol containing compounds, which does not show the red or pink color, whereas the Millen's positive test answers for the tyrosine or phenol containing compound with the red or pink color precipitate. Biuret test. To 2 ml of filtrate, add 1 drop of 2% copper sulfate solution, and to this add 1 ml of ethanol, followed by excess of potassium hydroxide pellets. Purple color indicates the presence of protein. Biuret reagent is an aqueous solution of potassium sodium tartrate treated with cupric sulfate and sodium hydroxide. The compounds with peptide linkage undergo the Biuret test. Proteins are polypeptides of amino acids linked together by peptide bonds. This is the diagrammatic representation. The Biuret test gives a purple color, and whereas the negative test gives a blue color. Xanthoproteotic test. The 3 ml of extract, add 1 ml of concentrated sulfuric acid. A white precipitate is produced, which turn yellow on heating, and orange addition by adding ammonium hydroxide reveal the presence of proteins. Xanthoproteotic test is used to detect amino acids containing an aromatic nucleus like tyrosine, tryptophan, and phenylalanine in a protein solution, which gives yellow color nitro derivatives on heating with the concentrated nitric acid. The aromatic benzene ring undergoes nitration to give the yellow color product. And this is the diagrammatic representation. In the starting, they will be of yellow color, and the end product will be a red color. Ninharin test. To 3 ml of extract, add 3 drops of 5% ninharin solution and heat it in a boiling water bath for about 10 minutes. Appearance of purple or bluish color reveal the presence of proteins. Proteins react with the breeding solution of ninharin and change to a color solution from a deep blue to violet pink, or sometimes even to a red color. This is the diagrammatic representation of ninharin test, which gives first the negative ninharin test where amino acids are absent without any color, and the positive ninharin test where amino acid present will be purple color complex and proline gives an yellow color precipitate. Next comes the amino acids. Introduction for the amino acids. They are building blocks of proteins and are linked in series of peptide bond to form primary structure of a protein. Amino acid possess an amine group, a carboxylic group, but a varying side chain that differs between different amino acids. There are 20 naturally occurring amino acids which vary from one another with respect to their side chains. Their melting points are extremely high, usually exceeding 200 degrees centigrade, and they exist as deuterions rather than unionized molecules. Amino acids respond to all typical chemical reactions associated with the compounds that contain carboxylic acid and amino group, usually under conditions where the zwitter form of ions is present in only small quantities. All amino acids except glycine exhibit optical activity due to the presence of an asymmetric alpha carbon atom. Amino acids with an Leva configuration are present in all naturally occurring proteins, whereas those existing in dextra form are found in antibiotics and in bacterial cell walls. Test for amino acids. First test, Ninharin test. To about 1 ml of amino acid solution, add 5 drops of 0.2% Ninharin solution in acetone. Boil over a water bath for about 2 minutes. Allow to cool and observe for the blue color formed. In the pH range of 4 to 8, all amino acids react with Ninharin, a powerful oxidizing agent, to give a purple color product termed Reuman's purple. All primary amines and ammonia react similarly, but without the liberation of carbon dioxide. The amino acids proline and hydroxyproline also react with ninharin, but they give a yellow colored complex instead of purple one. Next test is xanthoproteotic test. To about 2 ml amino acid solution in a boiling test tube, add equal volume of concentrated nitric acid. Heat over a flame for 2 minutes and observe the color. Now cool it thoroughly under the tap and cautiously run sufficient 40% of sodium hydroxide to make the solution strongly alkaline, and yellow color is produced. Aromatic amino acids such as phenylalanine, tyrosine, and tryptophan respond to this test. In the presence of concentrated nitric acid, the aromatic phenyl ring is nitrated to give yellow-colored nitroderivatives. At alkaline pH, the color changes to orange due to the ionization of the phenolic group. Next is Millen test. To 2 ml of amino acid solution in a test tube, add 1 to 2 drops of Millen's reagent. Warm the test tube in a boiling water bath for about 10 minutes. A brick red color is a positive reaction for the phenolic amino acids. Phenolic amino acids such as tyrosine and derivatives respond to this test. Compounds with the hydroxybenzene radical react with the Millen's reagent to form a red color complex. Miller's reagent chemically is a solution of mercuric sulfate in sulfuric acid. Next test is Sagakuji test. To 1 ml of sodium hydroxide, add 3 ml of arginine solution mixed with the 2 drops of alpha naphthol. Mix thoroughly and add 4 to 5 drops of bromine solution. A red color will be observed. 1% on naphthol in alcohol with a few drops of 10% sodium hydrobromide solution of bromine water and 40% of sodium hydroxide is Sagakuchi reaction. Under alkaline condition, alpha naphthol reacts with mono-substituted guanidine compounds like arginine, which upon treatment with hypobromite or hypochlorite produces a characteristic red color. Then lead sulfide test. Lead sulfide test undergoes boiling of 2 ml of amino acid solution with a few drops of 40% NaOH for 2 minutes. Cool it and add a few drops of sodium plumbate solution. A brown color or precipitate is positive test for sulfides. Sulfur containing amino acids such as cysteine and cysteine upon boiling with the sodium hydroxide yield sodium sulfide. This reaction is due to partial conversion of the organic sulfur to inorganic sulfide, which can be detected by precipitating it to a lead sulfide using lead acetate solution. Then comes the test for steroids. Lieberman's-Burger test. The amount of 0.5 gram of extract was dissolved in 10 ml of anhydrous chloroform and filtered. The solution was divided into equal portions for the following test. The first portion of the solution above was mixed with 1 ml of acetic anhydride, followed by addition of 1 ml of concentrated sulfuric acid down the sides of the test tube to form a layer underneath. The test tube was then observed for green coloration, which is an indicative of steroids. Another test is called as the Salkovic's test. The second portion of the solution was mixed with concentrated sulfuric acid carefully so that the acid formed a lower layer and the interface was observed for a reddish brown color indicating a steroid ring. Thanks for patient hearing.